of this year. For those of you who don't know us, ECUSA stands for Spanish Scientists in the US, but we welcome not only scientists, but also people working in technology, science and technology teachers, and people working in communication and administration in the USA. Our goal is to bring all of you together so that you can help each other and establish collaborations. We also want to expose the high impact work developed by professionals working in science and technology. And we want to be a point of contact for the Spanish and American institutions, as well as the media. To fulfill all these goals, we are working in a program for this year where we will have some seminars related to professional development. In fact, we will have a workshop in February about how to improve your skills in communication. And in March, we will have another seminar related to networking. We will send you all the information if you're interested. We also will have some seminars related to science and technology during this year, like the seminar we are holding tonight. We will organize some networking events to bring all of you together. And we also have a program focused on education. It's called Science in the School. And here we want to bring volunteers to the schools to talk about their work in science and technology. And last but not least, we have the International Mentor Program. Here we have volunteers. They are originally from Spain, but are currently working here in the US, and they will help and guide students who are in Spain via Skype. In fact, Maria is here. She's one of the organizers of this program. If you want to talk to her later during the reception, she will be happy to help you. If you are interested in any of these programs, if you want to be a volunteer, have any other ideas, suggestion, anything, please contact us in this email address, boston@ecusa.es. We also encourage you to follow us on the social media. We are on Facebook, Twitter, and LinkedIn. And we also encourage you to become a member of ECUSA. We'll send you a link to do it. It's very easy. It only takes a couple of minutes. This is the event that I told you about communication. We will have Dr. Rafael Luna coming to the Instituto Cervantes in February 18th. And I have personally attended one of his seminars, and it's highly recommended. It really improved my skills to communicate myself. So we'll send you the information after this seminar. And tonight, we have here Gonzalo Giribet. And I would like to thank not only him, but also the sponsors, Fundación Ramón Areces and FECIT, Spanish Foundation for Science and Technology, for supporting this event, as well as the Real Colegio Complutense for helping us with the logistics and donating this room. Gonzalo is here tonight to talk about how to use invertebrates to study evolution. He did his undergraduate at the Universidad de Barcelona at the Department de Biología Animal. From there, he moved to the American Museum of Natural History in New York, where he did his postdoctoral research in the laboratory of Dr. Ward Wheeler. In January of 2000, he started his own laboratory as an assistant professor in the Department of Organismic and Evolutionary Biology, as an assistant curator of invertebrates in the Museum of Comparative Zoology. From there, he was promoted to professor in 2007, and in 2012, he was named Alexander Agassiz Professor of Zoology in the Museum of Comparative Zoology. As an evolutionary biologist, he's interested in the origins of animal diversity, and he uses invertebrate animals as model to understand ancient biogeographical events and connectivity among populations. Gonzalo, thank you again for coming tonight.
Is this okay with the microphone? So I guess the talk, they said it was going to be in English, so everyone is fine with that, or do people prefer Spanish? Or okay. So I just and this is going to be a very unusual talk for me. I normally don't give talks to people who don't work on similar topics. So I'll try to do my best to showcase the research that we do in my laboratory in the Museum of Comparative Zoology, and also some of the interesting things. You know, some of them have to do with traveling around the world looking for the most beautiful creatures out there. So this is just a map. It's actually a little bit old. We could cover much more, but this is a few years ago. I had to give one of these talks to some high school kids, and I was showing where we had projects at that time in the world. So our research is global in the sense that we have to go and look for the animals that we need to work on in many different parts of the world. And there's different reasons for that. One of them, as I will show, is biogeographical. We want to study the organisms that live in different continental landmasses and how they've been changing as the continents have been reconfiguring. And other times we have to go to places where there's animals who only live in that particular place. So if you get unlucky, you may need to have to go to New Zealand to collect something that only lives in New Zealand. You know, it would be much easier if they live in our backyard here, but you know, the winters are a bit tough. I tend to work on things that are a bit more tropical, so they don't survive well. So we work on invertebrates, and I know that most people are familiar with some invertebrates. I just want to tell you a little bit. This is a pie chart of the total animal diversity. If you count all animals in the world, you'll see that most of them, in fact, about 96% of animals are invertebrates. The other minority, about 4%, are the vertebrates, things that you might be familiar with, like whales, T-Rexes, or yourself. Okay? But most animals are invertebrates. And in fact, when we look at them from an evolutionary perspective, we can see here a tree, an evolutionary tree that is trying to depict the relationships between those different animals. And basically, all the animal tree is invertebrates, except for very small, insignificant branch here that includes us. Okay? But most of the important animals are invertebrates, the ones that have interesting morphologies, interesting biogeographical stories, and that's what I study. And in the lab, we actually cover most of the diversity of this tree. We've had people who work on sponges or corals or all types of worms and all types of arthropods. And I'll show you some examples of the work that we do and why we do that type of work. One of the things that we do in the lab, and it was in the title of the talk, is biogeography. Biogeography, to me, is a very appealing branch of biology because it's really integrated. You have to incorporate information from evolution, from geology, from Earth history, and you have to look at things with many perspectives in mind, from ecology and other aspects. So it's, you know, I will say that biogeography is the science that attempts to describe and explain the patterns in the distribution of life on Earth. Okay? And we can do that from different perspectives. One is a historical perspective. That's the one that is going to explain why there aren't polar bears in Antarctica, for example. I mean, ecologically, they could live there, but bears evolved in the northern hemisphere. They never crossed the equator, and therefore they never got to Antarctica. Right? So that's the historical component. We're trying to understand some of those aspects. And there's an ecological component that I don't work on that as much. It's less evolutionary, but it will be just also assessing whether the environment is amenable for some organisms to live there. Okay? So I'm going to focus a little bit on some of the Earth history aspects and some of the more historical components of biogeography, but I think that it has, you know, these great properties of integrating a lot of different parts of biology, from ecology to evolution and other disciplines, especially Earth history and geology. One of the figures that I'm trying to follow somehow is Alfred Russel Wallace, you know, the father of biogeography. He spent many years in his life navigating the Rio Negro in Brazil, or, you know, collecting birds and butterflies in Southeast Asia. He was a true zoologist at the time, where he would just leave home and come back 5 years later full of specimens and things like that. And he was known, among other things, for proposing at the same time as Darwin, and in fact, it triggered Darwin to publish his Origin of the Species when he got a manuscript, a letter from Wallace saying that he had reached these conclusions from looking at this biodiversity around the world. Darwin had been working on that for many years, and that both of them presented an original paper in the Royal Society of London, in the Linnean Society of London, sorry, where they actually announced, the origin or evolution via natural selection. He was an amazing person. The other, so one of the aspects that at the time of Wallace and Darwin we didn't know are some new developments that happened during the 20th century mostly, and it's the theory of continental drift and the explanation of plate tectonics. And continental drift is basically a theory that says that the crust of the Earth is moving, and with that, it's taking the continents and changing their configuration. And this is something that we'll see was proposed by a German meteorologist, although he didn't have the mechanism to explain that continental drift. And then the plate tectonics explains actually how that continental drift. So those two aspects were instrumental developments from geology, from study of deep sediments in marine biology, that actually allowed us to really understand evolution or bring it to a different level. This is Alfred Wegener, and he proposed this idea. Everyone thought he was completely crazy, saying that the continents were not stable. They actually were moving, and they were moving quite quickly, and that's how you could line up the map of South America with the map of Africa, for example, and those things have been separated. And actually, he died in his last expedition when he went to Greenland, because he postulated that Greenland was the landmass that was moving at the fastest pace. He went to Greenland to take some geological measurements to prove his theory of continental drift. But obviously, then later on, people started understanding continental drift and understand that what we see as continents are only the emerged part of some plates, these tectonic plates, and they're always in contact. And obviously, in the areas where we have those tectonic plates, that's where you have all the natural disasters like, volcanism and like earthquakes. So the reason why, there's those big earthquakes in San Francisco is because it's in the verge of two plates, and when that plate moves, it creates those earthquakes. And the reason why there's so many volcanoes and things like that in Southeast Asia is because they have this thing that is called the Ring of Fire, which is like two plates, super active, and there's a lot of volcanism, there's a lot of tectonic events. And now we're beginning to understand how these things actually happen. And once we see these plates, we can understand that new crust is being formed all the time in the middle of the oceans. So you go to the middle of the Atlantic, there's the oceanic ridges where actually the lava is coming out, and there's forming new, it's adding new material to the crust. And as this lava emerges, forms volcanoes, and then this is moving towards the continent. And then since it's heavier than the continental plates, then it just goes inside in what we call, the lithosphere goes there in these oceanic plates, and that's that's a continuous movement that it continuously reshapes all the continents. Okay, so nothing static. There's land being added all the time in the middle of the oceans, and in fact, when you look at a map of the oceans in the world, you see that all this red means very young, from 0 to a few million years. So when you go to the middle of the Atlantic Ocean or any other oceanic ridges, what you see there, the rocks that you see there are very young. And as they spread towards the continents, they become older and older. But the oceans in general are actually pretty young, because once they reach the continent, then they go underneath. And most oceans, you can see they're only about 120, 150 million years. But we have actually many rocks in the continents that could be even a billion years old. And there's only one ocean that is really, really old, and it's the Mediterranean. The Mediterranean has a lot of old material deposited there from the old Tethys that you probably all have heard of. And the oldest oceanic rocks are about 280 million years, while many, as I said, many of the other rocks and continents, they can be Cambrian, Precambrian, things like that. So once we start understanding that, what I'm interested in is the organisms that live there. And I look for really old animals that could be 400 million years old, 300 million years old. And what I want to do is to trace them back. You know, if you go now back in time to, you know, about 140 million years, when the continents look completely different, and Africa and South America were still together, and together with Antarctica, and Antarctica wasn't frozen, and you had an entire fauna that lived all over these southern continents. This is the famous Gondwana. You had dinosaurs in Antarctica, and you had a lot of faunas that nowadays are found typically in southern South America, obviously not in Antarctica because it froze after the opening of the Great Passage. But many things you find in southern South America, southern Africa, Madagascar, India. This is now underwater. That's the Kerguelen Plateau, but then Australia and New Zealand. Okay? So one of the things that we do is go to all these land masses and look for animals that live there, and then bring them back into the lab and trying to study their evolution and see whether we can trace the history of how these land masses broke up by looking at the organisms that live over there. And you can come up with maps or trees like that tells you that, you know, Australia and New Guinea were connected until about 30 million years. Okay? Or that, for example, South America, where South America separated from Australia and New Guinea about 52 million years ago, and it separated from New Zealand about 80 million years ago. And once we have some of these points, we can start looking at what happened with the faunas. You know, is the fauna of New Guinea more closely related to the Australian one than it is to the one in South America, etc. To do that, I use typically, well, different groups of organisms. So one of my favorite ones are these things called daddy longlegs in English, or harvestmen. The scientific name is Opiliones. In Spanish, we call them Opiliones. And they're very interesting because we have fossils that are about 410 million years old. So the group is really old. A lot of its diversity originated pretty early. And from the Opiliones, I've been working mostly on a group called Cyphophthalmi. And you have a few individuals on the ventral view there. They all look pretty similar. But when we go to all these places, we find that they have very conserved biogeographic patterns. So for example, these are all specimens from Southeast Asia. You know, Southeast Asia, just they colonized Southeast Asia at one point in time, and then, you know, they diverged. Or we have another group that is found in West Africa and Southern South America because they were connected only about 100 million years or something like that. And we have another group, this one in New Caledonia, which is pretty difficult for us to explain biogeographically the origin of New Caledonia, especially because it was probably submerged about 52 million years ago also, and then re-emerged. So it's a bit more complicated. But then we have a typical group, a Holarctic group from the Northern Hemispheres found in Europe, North America, and Asia. And then this other group, which is the one that we can put in most emphasis, this family Petalidae, that is found, as I showed you earlier, in Chile, in South Africa, Sri Lanka, Australia, New Zealand, and Madagascar. So the group is perfect for doing these types of studies, and with that, we can actually try to trace back how those land masses broke. And, you know, I'm not going to pay much more attention to that, but just show you that you can, you know, go back in time to about 65 million years ago and see that, yeah, this group that is now found in Africa and South America, I mean, 65 million years ago, it was connected. So if the group is old enough, you can explain these patterns that we find nowadays by a process that we call vicariance, which is the animals stay there, they don't move around much. It's just as the continents separate, they end up having these disjunct distributions. And if you go even farther in time, as I showed earlier, all these things that are in New Zealand, Australia, South Africa, Madagascar, Chile, you know, they were connected. They had probably a much broader distribution. They were for sure found in Antarctica. You know, it's hard to demonstrate because you can't go to Antarctica and find fossils from, you know, below kilometers of ice sometimes, but we see that these distributions make a lot of sense once we explain the age of the organisms and go back in time and look at the configurations of the continents. And if you keep going back, same thing for, when Europe and North America form part of this larger, or the northern portion of this supercontinent called Pangea. Okay? So this is some of the things that we're trying to do, and we're looking at, we have to go to all these places, sometimes they're not very convenient, and find some of these animals by, mostly the animals that we look at, they live in the leaf litter. And the reason for that is that this is a very stable environment. These animals normally don't disperse well. They need to be in high humidity, in undisturbed forests. So we go to these places, we use these sifters, put a bunch of leaf litter there and get a lot of the tiny creatures that live in those places, which are the ones that then we bring to the lab to do some of these studies. And we look at other animals also. On top there, we have a couple rhicinulates, which is another group of arachnids, very interesting. Nowadays, they're only found in the neotropical region and in the West African region, but in the past, they were also found in Southeast Asia, so they probably had a much broader distribution, and they went extinct at the time. We look at things like centipedes and other weird arachnid groups like these ones called schizomids. And these are things that I like to collect. They're all small, they all live in the leaf litter, they all have these narrow ecological requirements that allow them to, or don't allow them to disperse very well. We look at other things also, but mostly a lot of the leaf litter animals. And one of my favorite leaf litter animals that we've been working on lately are these things called velvet worms or onychophorans. A really cool ancient group. We have fossils from 310 million years old that they look exactly like these guys. The distribution nowadays is a bit restricted to the south, tropics and then southern continents mostly. But their ones were also much broadly, distributed much more broadly because they were also in the northern hemisphere. There's fossils from France, for example. And we've been doing the same type of work. When we look at the phylogeny, the relationships between the individuals that we collect, we find that we have a tropical family that was found in, or it's found, sorry, in the tropics, West Africa, and also Southeast Asia, which is the same distribution that those other rhicinulates had, for example, 100 million years ago. And the other group has the same distribution as the leftmost cyphophthalmic clade that I showed you. They're found in Chile, they're found in South Africa, Australia, New Zealand, and also New Guinea, for example. And when we look at the resolution of these things, we see biogeographical patterns that are very common. For example, finding Tasmania and New Zealand being related. Tasmania is this little island here in New Zealand. They have a lot of shared fauna between these two parts. So we're able to trace back a lot of these relationships by looking at the animals that live in those land masses, and we hope to be adding more and more species to these types of analysis. We also do other type of work, very different. This is easy field work. You go, to different parts of the world. Normally, you rent a car or hire someone to drive you around, and small teams. But some of the work requires a lot of logistics and large vessels. Like this expedition, we took the largest oceanographic vessel from the Spanish government, the Sarmiento de Gamboa. And we went to this expedition in 2009 to look at the Banco de Galicia, which is an area that reaches down to about 5,000 meters. It's in Banco because it's a fisherman's area. That's where they get a lot of the fisheries in Galicia. And it's located about 8 hours navigating west of Vigo. And there were several things that they wanted to do there. One of them, they wanted to find the geologies whether there was granite in the Banco de Galicia. The reason for that is that they found granite over there. They could claim that that is part of the continental shelf and that therefore it belongs to Spain. If they don't find granite, it means no, it's part of the common ocean. You cannot claim fishing rights and all that. So one of the intentions of the geologists was to find granite. These expeditions are very different from the other ones, very expensive vessels, large teams of people doing all different types of work. And also, hiring a boat like that costs a lot of money. Somebody pays for it, I don't know who, but somebody pays many thousands and thousands of dollars. So you work 24 hours. So what you do, divide the crew in shifts, and you work in shifts of 6 hours, or 8 hours, and then you work 8 hours, you rest 8 hours, you rest 8 hours, you work 8 hours again. No, 6, sorry, because you do 1.5 shift every day. Some nights you don't sleep, some mornings you're working. It has much more, obviously, safety things. We have to test, the suits in case the boat sinks, and there's a director. This is our team, and this team was composed by people from Universidad de Vigo, Universidad de Santiago, from a museum in Munich, and then from myself from here, from Harvard. And there were people from other international people in other ships. And this is the maximum depth that we went. So this is very interesting because working at these depths, has a lot of logistics. And just 5 kilometers down the surface of the ocean. What's the problem with that? Well, the problem is that when you send something down there, you might take 4 hours to send your dredge. You don't know if it touched bottom or not because you have tons, like real tons, thousands of kilos of wire, which is like moving with underwater currents. You wait like 2 hours thinking that you might be catching something, and then it takes 6 hours to bring it back up, and maybe it's empty, maybe you've lost a gear. So it has a lot of logistic problems. And we're doing, testing different techniques, different dredges to collect. This is one special dredge that is trying to collect only the animals that live on the surface of the bottom. So when this touches the bottom, it disturbs them, and then they come here in this fine mesh, and you collect animals that, they're not inside, but they're just like swimming around on the surface. And we're testing many different things, like a lot of the phytologists or the geologists wanted samples of water, so you can take these nesting bottles at different depths and open them there, and you collect water from 5,000 meters or from 2,000 meters and things like that, and also the animals or the plants or algae that live at those depths. This is the dredge that the geologists really wanted to test. Is la draga de roca, the rock dredge. It's just a huge dredge. It just breaks anything that it touches. Either breaks the rock or you just lose everything. It's either one. It's pretty heavy, and the idea is that that you bring pieces of rock here, and then you can examine what types of rocks you have in the bottom of the ocean. And there's other things, the logistics here, for example, is a device that we send down the water, a very expensive dredge that was designed for this expedition. This is made of like really thick inox, but the designer forgot to drill little holes along here. And obviously, when you bring something that has air to 5,000 meters depth, it just collapses it like if it was paper. You can see what happens to a normal, styrofoam cap when you put it at those pressures. It just compresses. So this is very typical that people send a few of these things down with drawings and all that, and then you get these super compact little ones. You can see here, this was the original diameter of this stainless steel, really thick. It just came like flat, completely flat. So that one didn't work anymore. And then you're working many hours, ships in the boat, sometimes rocking really badly. Like the last 2 days, we had very bad storms, and you're trying to get the dredges to work, the animals, photograph, fix, prepare for the analysis, and then bring them to the lab. But you get things that are pretty cute. Others are pretty obscene looking animals. All sorts of things that live in the bottom of the ocean. These are some on it. So it's really interesting fauna, and the idea is to go there to collect things that obviously will not be accessible with other types of expeditions. So deep sea expeditions require a lot of logistics, but you get animals that are very difficult to obtain otherwise. And what we do with a lot of these things is a lot of DNA work. And in fact, we do a lot of genomics or transcriptomics in the lab. Some of the lab members are here. They know much more about the transcriptomics than I do. But the idea is to bring these things and try to get as much genetic information as possible to draw these evolutionary trees, to look at the biogeographic questions or look at the evolutionary questions. So, and I think that you all know a little bit about DNA and the importance that in 1953, they discovered, they published that seminal paper by Watson and Crick, where they deciphered the secondary structure of the double helix, the tertiary structure of the double helix. Took a long time until people could really do much stuff with DNA. I mean, they knew the structure, but you couldn't do much with it. But in 1977, Frederick Sanger invented the method that was still used, many people still use, to actually read the DNA code, which is a method for what we call sequencing DNA. And this method, he developed some enzymatic reactions to stop replication of DNA and be able to read it. And then a few years later, people started using this thing, reading the DNA sequences, to compare DNA sequences between different animals to start coming up with the first trees of animals. Okay? And we use now DNA routinely to give, this is the original paper. Obviously, that original phylogeny was based on very little data from a single gene, and the relationships are not what we think nowadays. But that was, the seminal first paper where they actually used DNA sequencing for looking at evolutionary trees. And from then, things went really quickly now. From 1988, the first animal phylogeny. In 1996, you have the first eukaryotic genome for fungi, Saccharomyces cerevisiae. 2 years later, the first animal genome was published, only 1998, using Sanger sequencing, brute force type of thing, hundreds of labs, sequencing. In 2000, you have the first Drosophila, the biggest model organism, also the biggest plant model, the first plant genome for Arabidopsis. Next year, it's the sequencing of the human genome. And there were the two competing papers, the one in Science from the consortium that had hundreds of labs all over the world sequencing a lot of these things. And then Craig Venter came a couple of years later, said, I'm going to do that on my own with my own company. And he actually, in 2-3 years, was able to sequence the human genome. And that one was published in Science. So they had one in Science, one in Nature, each one. But things started changing a lot. There were now these massive places to do sequencing, still using old technology, but brute force, lots of machines. But what we had seen until then, until 2007 more or less, is that sequencing still follow this Moore's law, which the bioinformaticians or the computational people know what it is. You basically are able to duplicate the number of transistors of one motherboard every 2 years. So it's not a very sustainable way of sequencing. The first genomes cost about $100 million. By 2007, we're about $10 million per genome. Obviously, the lowly zoologists like me couldn't even think of sequencing that, and that's why we only had a few genomes from a few model organisms where there were a lot of resources invested in. And we had the mouse, and we had humans, and a few Drosophila, C. elegans, a few other things. What happened after 2007, it's key for a lot of the research that we do nowadays. And is the development of new technologies for sequencing, completely changing the paradigm of this Sanger sequencing, and people started using other machines. And here, just showing like the Illumina machine, which is one that we routinely use in the lab, where you're actually able to sequence hundreds of millions of sequences for just a few $1,000s. Okay, and this is now we're talking about possibility of using these technologies to sequence entire genomes for a few $1,000s instead of talking about millions of dollars. And this is what we've seen since 2007. Then sequencing capacity, price per base just went down, and we're now, at a limit of, you could do a sequence, an entire genome for about $5,000, and these prices keep going down. I mean, in fact, the goal is that within the next couple of years, people are thinking about sequencing entire genomes for $100 to $1,000. And once we're able to do that, we can do anything that we want in evolutionary biology. There's no model organisms anymore. We can use anything that we want. So where's this technology taking us? And this is the last 5 minutes of the talk. They told me about 30 to 40 minutes. So this is obviously for us, is putting us in a great position. Before, we could only access to these model organism genomes and then use those to compare to our organisms, but really not be able to do the same type of work. Now you can do, a couple of years ago or last year, no, 2010 already, yeah. So they discover in Siberia these a couple bones, like fingertip bones and a couple teeth from a hominid that they didn't even know what it was. They had no idea how it looked. They knew it was hominid bones, and they were able to sequence from this fossil bone an entire genome. This is how you can, where you can get nowadays with some of these technologies. And this, again, didn't require millions of dollars. This was only thousands of dollars, highly fragmented DNA from an hominid that they discovered is an entire new lineage. It's not Homo sapiens, it's not Homo neanderthalensis. So you can get there. And obviously now there's large consortia of people who are there, for example, the I5K. This is a community of insect people who are trying to sequence about 5,000 genomes of insects. They haven't sequenced the 5,000 genomes, we must say, but very recently they published a beautiful paper in Science where they had transcriptomes and genomes for a large number of insects and are able to provide very complete pictures of the evolution of these very large groups of organisms. And there's many other initiatives with plants and with nematodes, and we have our own that is called GIGA, which is the Global Invertebrate Genomics Alliance. And this is trying to bring community resources and expertise from different people to actually contribute towards the genomics, not necessarily genomes, but also transcriptomes of as many invertebrates as possible. I mean, these initiatives are beginning, they don't have a lot of funding, but now the funding is coming from a lot of the private labs. So what we use this data for is to infer the tree of life. And I'm not going to show you many trees, but I just want to give you an example of what we can do with that. And this is something, some of the research that I was involved with a couple of years ago, where we actually found that a group of animals called ctenophores were actually found to be the most basal animals, more basal than sponges. And sponges were thought to be basal because they don't have a nervous system, they don't have any tissues structured in the typical way that we know in other animals, but ctenophores do have those things. We did this phylogenetic analysis using large amounts of data, tiny compared to what we use nowadays, but the idea is that we found that the ctenophores are at the base, so more basal than porifera, and the alternative option is whether the sponges are at the base. This is a debate that's still going on in the literature, in the community. We're trying to generate more resources to answer questions like this. Are ctenophores the most basal animals, therefore implying that all animals originally had a nervous system and a tissue organization, and then it was lost in some lineages like sponges and other lineages like placozoans, or were simple animals without tissues and without nervous systems first, and then complexity evolved later on. So we're also trying to answer other similar questions, like, for example, what is the base of the bilateral animals, the animals that have bilateral symmetry like we do? There's some basal animals that don't look like us. They're not cephalized. They don't have bilateral symmetry. They might have radial symmetry. They might be asymmetric like many sponges. So there's an idea that at the beginning, at the origin of these bilateral symmetries, there were some animals that are called aseals or cenoturbelids that don't have a through gut. The gut is not well developed. They don't have a brain like ours. They do have some concentration of neurons in the anterior part of the body, completely disconnected from the mouth. We always associate cephalization with two functions. One is sensorial and the other one is feeding. But many of these things, imagine an animal that has actually some brain in the anterior part of the body and the mouth located, in the posterior part of the body, completely separated. So there's also some debate about the origin of the bilateral animals and how that evolved. And one of them is whether these basal animals go early in the bilateral tree, or they're actually more closely related as very simplified, secondarily simplified animals, but they're related to things like us, the Eurostone, things like sea urchins and vertebrates and things like that. So those are the types of questions we're trying to answer. For a long time, we've been limited in the amount of data we could throw at these questions. We had only a few genes that we sequenced via Sanger sequencing, or the first technologies for generating some of these larger data sets. And what we're doing nowadays is actually trying to sequence entire genomes or very complete transcriptomes for many animals, also to try to address some of those questions. And then we come up with, trees that look like that. This is some of the last complete, more or less complete animal phylogeny, which we published, many years ago now, in 2009. No one has really looked at this with all the new technologies. This was used still with that technology that I showed you up to 2007. And what this generated, I'm going to skip that, are matrices that are fairly incomplete. This is our 2009 matrix, and we're looking at about 1,500 genes for all animals. And you can see that on top you have some animals for which we had genomes like Drosophila, C. elegans, or humans, but we had a lot of genes, a lot of black. But many of the other ones, they had very sparse sampling, just a few gene sequences here and there. Nowadays, we can generate matrices of the same size. This is a matrix we're analyzing for worms, for annelids, has the same number of genes, about 1,473. But you see that here most of that graph is black. Okay, so this is the change in technology has allowed us now to be able to generate the amounts of data that we had in 2009 only for model organisms, but now we can do it for pretty much everything. Obviously, this poses a lot of analytical challenges. A lot of resources in the lab are now spent just in the computers and fighting who's going to get all the CPUs this month. And Rosa is running out of karma all the time. Karma is the way they used to measure how much work you've been doing in the common cluster. And she burns it all the time because they're all, we're always doing these types of analysis. The idea is to come up with complete trees. This is one tree that we're getting ready for publication, but we're including the first transcriptomes of really small animals that are about 125 microns in size, for which we only had like 1 or 2 genes. And now we have these very large amounts of data. We can really evaluate the position of enigmatic animals like these other 2 here, Dirodrilus or Lobatus cerebrum. These are two animals that no one knew what they are. They're always called problematica. People didn't know whether they were related to annelids or to flatworms or other things. And with these amounts of data now, we have pretty strong results suggesting that these very enigmatic animals actually come with annelids. So this is the types of things that we're trying to do with this data. When we're trying to look at the tree of life, the earlier data, when we're trying to see how continents move and the faunas that live there, is more of the biogeographical work, but it is a lot of fun. It's great because you have to interact with people with expertise in many different areas, from the animals to the field to the computers, genomics, and that's really good. So with that, I just want to conclude by thanking the lab. And this is a photo taken from the lab outside of our building. Some of the funding sources, especially the ATOL Assembly the Tree of Life program from the National Science Foundation, and also from some of my collaborators that have been instrumental in some of the work that we've been doing in these aspects through the years. If there are any questions, I'll be happy to try to answer.
Questions? I have a question.
Yes.
So I'm just curious about how do you decide that you're looking for when you look something for a species, and then you trace down from the fossils, or when you first start thinking, okay, I'm going to gather a species and characterize them, where to start?
Yeah, I mean, you obviously go to the literature and know that someone found some specimens there. Sometimes we have museum specimens. You go to the database, oh, we have this species from New Zealand, from here, from there, and you go there. Many times you take the risk. For example, I showed a picture of that velvet worm from Gabon. So that had only been collected, there are 3 papers on that animal, 2 of them from 1890 something, another one from 1906. No one has talked about that animal anymore. It's the only onychophoran of that family in entire Africa. So, I went to the area that it was described, and you look for forests that are more or less still decent. In some of those areas, the forests are gone. And then once you find those forests, you look for the habitat for those animals. and look. In that case, we were in West Africa for about a month, and the last day in the field, we found one specimen. So it's, yeah, we picked it up. Yeah. We're not going to leave it there for the next person. So it's risky, and one of the things that I always do, because I know the risk of some of these things, is that I don't plan a major expedition like that for just one thing. You know, there's always multiple projects involved. So if you fail at one, at least you have others. In this case, we found our 4 main goals. We found it. This one was pretty frustrating. We broke a lot of logs. You know, we look for rotten logs where they're just, you can break them apart, and we looked for a lot of logs before we found that animal. Might be the last one in the species. Who knows?
So do you need to know what they look like? I mean, like, you need to know all these species.
Yeah.
Or at least the
And that's, you know, that's where the students and the postdocs supersede you with the techniques and all that. But the many years accumulated of looking at the animals, that's the last thing that I can do now. At least, you know, it's just really know where to find them and how to look and teach the new generations too. Because a lot of people nowadays doing, especially people doing genomics, they don't go to the field to look for the animals. You know, it's not very common that people are combining those different aspects. Today I was filling some forms for the US government for something like that, and they asked how many days. I had to say how many days I had been outside of the US in the last 5 years, and I had it counted because I needed to count it for some issues, and it was like 600 days. And I said, they're not going to be happy, you know. But yeah, I spend every year from 2 to 5 months in the field collecting animals, and that's a very important. And not only me, and then there's people in the lab who go to other places. I think that if we add all the time that people in the lab spend in the field every year, it's more than 6 months. And that's what we have, animals that no one else has or will have. Yeah.
First of all, thank you for a fascinating talk. It was awesome. I think we are all jealous. We are switching fields now. No chemistry, no molecular biology, evolutionary biology. I was curious to know what drove you in the first place in terms of your career choice to choose this field. And if you could, I know Christina overviewed your career, but if you could walk us through how it was for you to come over here and give us a little bit of the personal perspective on that.
Okay, it is very nerdy. I mean, I was 4 years. I was 4 years old when I wanted to do this. You know, it's just like always. I always wanted to do that since I was a kid. I was collecting shells and insects and all that. And I always knew I wanted to be a biologist, a zoologist. I always wanted to work on these things. So that's a part of what I wanted to do. You know, most people want to be soccer players, and they're never soccer players, right? So, but I think that I was very passionate about it, and then it was, I don't know, it's almost as a game. It's fun, you know. It's just my, I always go to work unless I have the whole day of meetings, boring meetings. But every day, you go to work excited about the new research, the new discoveries, the new thing that I'm going to do. So in that sense, I think it's been easy for me, right? But then career-wise, I think that what is very important is to make decisions that are, that make you, sometimes people don't want to move, sometimes people, are more conservative. And I really wanted to go to New York to do my postdoc. I did my PhD in Barcelona, and I wanted to work with one specific person, and I work hard, and endure to go work with him. I remember the first time that he offered, yeah, you can come to my lab, but then I was emailing, respond and all that. And at the end said, look, I have this fellowship to come to your lab for 3 months. That was during my PhD. But if you don't sign this form today, I mean, I'm losing it. And then he signed it, and then I was there for 3 months, and I really loved the place, and he was very, very happy with the work I did. Yes, I worked very hard. He was very happy. He just the day I left said, well, if you want a postdoc, you have a postdoc here. So I think that, and that happens, I mean, I have a lot of colleagues that come to me and say, oh, I have this postdoc from Spain, just worked so hard and all that. We have a good reputation, I think, at least in the US, of people working very hard and being very well prepared, being smart. I think our university system is very good. It doesn't have some of the things that you have here, more the one-on-one, and perhaps not as much of the lab experience or the field component and all that. But I think we get pretty good training considering other things. You know, I think that research in Spain is getting better and better, but it's not there. But I think education at the university level, it's pretty good. It's pretty high standards. So I think that once you come here, the people who have drive, you know, people are happy, they're well prepared, and I think that then it's much easier to succeed.
Thank you for that. I was curious also about the funding, and I'm like, NSF, I guess that's a good fit. I was not sure what kind of agencies would.
Yeah, we only get NSF. The type of work we do doesn't qualify for NIH.
Or these more politically driven expeditions, right? Like the one in Galicia.
Yeah, well, but that's yeah. I mean that they invited me from one of the Spanish, you know, some of my Spanish colleagues who had the vessel. They invited me as a researcher. I mean, a lot of the things are done with small grants. A lot of the field work we do with internal small grants from the NCC, but obviously the big projects are funded by NSF.
What about in the museum? Given that you also work with them, is there any research that is subsidized or posted by the museum? Can you tell us a little bit about your work in the museum?
Yeah. So so the museum, I don't know if you know which museum it is. It's the Natural History Museum that is on Oxford Street. You might have seen the public face of the museum. The big part of that museum is actually the research side of the museum. The MCZ, which is my institution, is a very old institution here. And it has had a lot of tradition in working on zoology, comparative biology. So we have quite a lot of internal resources, and we pay for our students. We have internal grant competitions to do field work, and we have very large collections. We are the largest museum, university museum in the world. Most universities don't maintain the collections because they're expensive, and a lot of the universities who used to have museums, they've got rid of them or they're not supporting them anymore. And we're the only university museum that is comparable to the big museums like the Paris Museum or London Museum or the American Museum of Natural History or Smithsonian. So we're on par with these institutions, but we are a research university. So we have the curators of the museum were professors in the department and curators in the museum. So we have about 8 curators who take care of the different collections. My collection is invertebrate zoology, but obviously I'm not the one taking care of the specimens. I have the staff who works taking care of the specimens, databasing, sending specimens on loan, and all that. So they do the day-to-day work on the collection. I, you know, the scientific director of the collection. Yeah.
So how much genomics has changed the way we define species? Like, because I can imagine that a lot of them have been defined as by observation, or I don't know. Yeah,
I mean, traditionally people use morphology to define the species. Just look, you know, this one looks different from this one, and you know, to be fair, probably 90-95% of the times that's fine. You know, and it's the easiest way to tell apart two things. But then, you know, there's other times that you start looking at the genetics. For example, elephants. You know, when I was little, we studied there were 2 elephants, one in Africa and one in Asia. But when people started applying genetic techniques to look at the population structure of elephants, I mean, they realized that there's no connectivity between the populations of the forest elephants in the more tropical area and the open savanna elephants, which are larger. They're, you know, genetically, they're isolated, they diverged a long time ago, and now they're considered different species. Obviously, where you break what's a species or two sometimes can be arbitrary, whether you use genetics or not, but genetics has allowed to distinguish cases of cryptic species also many times, things that we think it's only one species, or the opposite. Sometimes we think that 2 animals or plants that look very different when you look at, their streams of their distribution. They say, oh, the one in the north is bigger and has larger ears, and the one in the south is smaller and just walks different within their different species. And then they look genetically, and there's actually a continuum, a genetic continuum across the entire distribution range, and you can merge those things. Okay, so it does help a lot. It has a lot of applications. People are using it more and more for defining species, but still, the old-fashioned morphology distinguishes most of the species pretty quickly. But I think that now we're trying to do more integrative taxonomy, where we're actually combining both things. Even when we describe species based on morphology, we tend to sequence a few individuals and make sure that they're all the same species. Genetics is also allowing you to identify things for which you don't have the characters. Let's say that you have eggs of insects or a dead piece of animal that was in the gut content of something else. You can also apply genetics to identify them. Well, you couldn't do it with morphology. So they're great complements to each other.
Can you go sampling anywhere? Do you always get the permission from government?
Not always, yeah. I mean, we always need the permits, yes, unless the country has no permits and they specify you don't need permits. We always need permits to collect everywhere. Some places are very difficult. I've never collected in India because getting permits for India is almost impossible. Indonesia is very hard. A couple of years ago, when we were going to Indonesia, the permits didn't come through, so we postponed that trip, and we actually had to go the next year. And some places they might not give you permits. But
why is that?
Sometimes misinformation about biodiversity laws. Brazil for a long time did not allow any genetic work outside of Brazil with their samples. They will not authorize anyone to take samples out of Brazil because what I call the Jurassic Park effect. They thought that you could take their DNA and reconstruct their fauna, with their DNA. Now, I must be fair, that they have a reason for it, and it's that Brazil is the country that has suffered most from biopiracy, and that was during the First World War. They actually had the rubber production, tires, goma. They produced the rubber for all the tires in the world, and a German went there, took some seeds of rubber plants, brought them to Indonesia, established plantations in Indonesia, and the Brazilian economy collapsed. So, they've been very careful with that. But there's this misconception of the, bioprospection, that you will go there and then patent the genes of their. So I think that now people are beginning to understand much more that everyone in science is using genetic tools, that you're not going to be able to clone their species. And they might have, some types of stipulations in the permits that the specimens, they need to go back there, or you're not allowed to do this or that with your specimens. So there might be some countries allow you to do morphology, but not DNA work. And these things evolve. Brazil was impossible. Now it's kind of okay if you collaborate with Brazilians. Colombia was impossible 2 years ago, but I think now they're changing their laws. And we're always playing cat and mouse with governments to try to get permits. I love New Zealand. That's why I go so much there. Last year we applied for permits, and they gave us 10-year permit to collect almost anywhere in New Zealand. Even my New Zealand colleagues are jealous. They're like, how come you got that? We cannot get that. I said, well, we've been working here for a long time. But sometimes you get things like that. Other times it's really hard to get permits.
I have a question. Well, first of all, I would like to second Marta, thanking you for this amazing talk. It was so interesting and very easy to follow. It brought me back to the days when I was studying zoology at Universidad Autónoma de Madrid. It was very interesting. So my question is, I guess there are groups back in Spain studying similar things, right? What is the major difference that you see between the research in Spain in this topic and here?
I mean, yeah, in Spain, there's a lot of groups now doing this type of work. I mean, Rosa, who's now in my lab, she comes from a group where they did some of this work. And the reason why she's in my lab is because she was doing great work there. Then, the difference sometimes are on, of scope and resources. one of the things that is very common in Spain, you know, they get grants often to do, let's say, work local areas or European fauna or something like that. It's very hard in Spain to get a project to do. Let's say you want to do your thesis in a very interesting evolutionary problem in New Zealand. It's very unlikely that you're going to get the funding to do that. And also, I mean, it's not only US versus Spain. I mean, you know, we're a harbor where we have a lot of resources. Not every university in the US has the resources that we have, unfortunately. And it's the same with Spain. Some institutes have access to a lot of these genomic tools, but many universities don't have access to some of these tools. What I think that is the biggest difference is the environment sometimes. It's, you know, it's who you have around or how open people are to talk to you, no matter how important they are. You know, I think that here any student can approach any famous professor and go talk to them or go have a coffee. I think that you're a bit more, it's much less hierarchical in that sense. And people know that if you're working at a university, it doesn't matter whether you're famous or not. If a student from that university wants to talk to you, you have to talk to them. You know, you have to use your time to talk to them. But, you know, obviously this is more like a personal thing. Some people might be better, some people might be worse. Some people might have had better experience in Spain than here. In my case, you know, when I came to the US, I found that I had so many resources to do things, and the people didn't hesitate to say, "Oh, this is great! You're doing a great thing." You know, I mean, no one ever said, "Oh, you're doing great." You know, because it's all I find, or I used to find, that it was always comparative. If I tell you, "Oh, you're great," means you're better than me, or no, great is great, and you can be great, and somebody else can be great as well. You know, it's not comparative as much, and people, you know, if you do something nice, people tell you. I think that's a bit different in that.
Do you have any collaboration with groups in Spain?
A lot
Yeah.
of them. And a lot of students from Spain come to my lab for short, 3 months or 6 months. Some come for 3 months and stay for 5 years. And you know, I have everything. And I've had a lot of photos from Spain. Yeah.
More questions?
Yeah, I have a question. Like, I really like that the way that you have explained, like the evolution of the field from the beginning to nowadays. So the question is that if in a few years there's gonna be the sequencing is gonna be available in most of the labs in the world, what is gonna change, or what questions do you think that is gonna be able to?
Obviously, you know, this is like everything. Probably the most interesting questions will have been, well, some will have been answered and some will never be resolved, obviously. This has happened before. When people started using DNA before, just, you know, what happened is that we started doing the big animal phylogenies, looking at the entire tree. And then once, you know, a few of us had done that, not everyone was looking at the same questions. And then they were looking at, you know, just one group of animals and then narrowing down to groups of species and all that. So it's a little bit, you know, an opportunistic thing. You get the big question first, you know, then the next person gets to the second big question. So I think that in that sense, being first doing these types of things helps, right? Helps me to get the money to continue doing some of that stuff. But there will always be things. I mean, there's millions of organisms. There's always things that we're trying to refine, whether it's taxonomy, whether it's trying to understand biogeographic questions, whether it's trying to understand ranges of species. This is something that we still don't really understand very well, you know, whether species live in all the oceans or they're isolated groups in each ocean. And we're trying to look at that with one example and another, but we still have a few million to go, you know. So I think that'll be always. And new discoveries. Last year, there was a new discovery of an animal that we have no idea how it fits that tree. So we have it in the lab now. It was collected from 3,000 meters about 30 years ago. So now we're going to try to use some of these genetic tools to actually see if we can recover highly fragmented DNA that was preserved in formalin. And we're trying, you know, we will try to use some of the techniques that people have used also for highly degraded DNA
to see if we can get genomes or fragments of the genomes of these organisms and place this new discovery in the tree. Morphologically, it's very different from everything else, so we don't really know where I mean, we know it's a basal animal. It's not something with complex tissues or organs or things like that, but we don't know where it fits the tree.
No more questions?
Let's have some.
Sure. We have some
Yeah, you can ask things there. You know, I'll hang around.
Another round of applause.